Is Francesco Tamagnini, who's going to talk about neural networks in dementia.
Yeah, if it can load. Wait a second. My name is Francesco Tamagnini, and I'm lecturer in pharmacology at the University of Reading School of Pharmacy. I'm a neuroscientist, so I'm not a mathematician, so I'm feeling quite intimidated by the whole lot of the formalism. But yeah, today I will talk to you a little bit about my work and how I try to, causally connect single channel alterations associated with dementia and whole network alterations, both for the development of new therapeutic strategies and hopefully for the development of also early diagnostic tools. But mostly I would like to have your feedback later on for how to actually make this happen based on my data. Or on the data that are being acquired in the field. So, as you know, Alzheimer's disease is associated to two main histopathological markers, which are the accumulation of beta amyloid protein, which as it accumulates correlates with the progression of the cognitive decline that manifests early on as a amnestic single or multi-domain mild cognitive decline, and then in time, amyloid beta leads to the accumulation of hyperphosphorylated tau protein intracellular tangles, and that's when the tauopathy, the so-called tauopathy, appears, leading to severe cognitive decline, which parallels with neural degeneration. Now, a fairly unvestigated field, a relatively unvestigated field, is how these different stages of pathology correlate with electrophysiological alterations and how these then are correlated or causally related with the cognitive decline that we observe, and how we can possibly harness this information to develop new therapies or new early diagnostic tools. So what we do know about Alzheimer's disease and altered excitability is that across Alzheimer's, along the progression of Alzheimer's dementia, there is an increasing risk in epilepsy or in seizures. We also know from preclinical models of amyloid beta pathology that we can observe this epileptic-like activity in the hippocampi, for example, of J20 mice, which are mice that overexpress amyloid beta species. And that, this pretty much informed the last 10 years of my work, give or take, which is, how do amyloid and tau pathology affect the excitability of excitatory neurons and inhibitory neurons, mostly in the hippocampal networks, and how these changes then account for the network alterations that we observe in animals, in animal models, and in human beings. And most importantly, so what? What can we actually do with this information? That's where I got stuck, personally speaking. So I hope to find some help in here. So briefly, what I do is to source genetically modified rodents that show signs of dementia. And I think Master Splinter is the perfect example of that. Then we kill these animals, we extract their brain, we keep the brain alive using artificial cerebrospinal fluid. Of course, the brain is big, it's got certain surface to volume ratio that doesn't allow nutrients to go in, so we section it quickly in 300 micron slices. These slices are thin enough to allow the free diffusion of nutrients, which are in the artificial cerebrospinal fluid, but thick enough to retain a certain degree of structural network integrity. Here you see two examples of a hippocampal slice from a mouse genetically modified to overexpress amyloid beta protein, so you can see the plaques being translucent in the entorhinal cortex and in the hippocampus, and you can see an example of a wild-type littermate. Then what we do is to use a technique known as single-cell electrophysiology, in this case patch clamp electrophysiology, and we use an electrophysiology rig to record the activity. The second part of the talk will be dedicated to clinical electrophysiology. This is a project that I carry out with Mark and Dom from the University of Reading, and so we're going to talk about that later on. So what I do with the single-cell electrophysiology data is to try to characterize alterations in the activity at different levels, starting from single channel, so I can run voltage clamp recordings to measure the biophysical properties of voltage-gated sodium and potassium channels. Then I can measure the excitability properties of single neurons, both the passive electrotonic properties of the plasma membrane and the electrogenic ones. And I can also measure spontaneous or evoked excitatory or inhibitory postsynaptic currents in these cells, so seeing how the local network works, right? And we can get a lot of information about each one of these levels, but we don't know very much how to causally connect them with each other. So what we do is to take the mouse, sacrifice it, slice the brain, keep the brain in artificial cerebrospinal fluid. We, then let it recover, and if it needs to be transported, we, can transport it in sealed Duran bottles. This is actually quite kind of cool because we came up with this protocol only recently, and we managed to find a way to preserve these slices for 3 hours without bubbling them. It can be useful logistically speaking. Then once we are in our lab, we use a micropipette filled with an intracellular solution that resembles the ionic composition of the neuron. We approach our pipette, which is mounted on a recording electrode, to the plasma membrane. We suck the plasma membrane inside in this configuration known as the gigaseal, because the access resistance of the pipette is in the gigaohm kind of field. Then we apply further suction and we break the patch of membrane between the cell and the pipette. So now the inside of the cell is in contact with the inside of the pipette, and this configuration is also known as as whole cell configuration, and this is the one that we mostly use. So in whole cell, basically we use a configuration known as current clamp, where we inject certain amounts of current and we track what happens to the plasma membrane voltage, right? So you can inject a negative current step and follow the single exponential decay. I said single exponential, this is as far as my maths go. Single exponential decay of the membrane voltage. This is fairly useful, yeah, because depending on how much this deflection, how big this deflection is, we understand by the Ohm's law that says that the voltage is equal to the resistance times the current, we can predict how much the voltage will change for the same amount of injected current, right? That's how synaptic transmission occurs, right? You've got a neurotransmitter, it binds a postsynaptic receptor, and it generates these small currents, right? It's particularly interesting when you're injecting positive current steps, driving the cell to fire. And you can measure the firing rates, you can measure the accommodation of the firing rate. That is, for example, very important for the coding of information, spatial information coding in the hippocampus. And you can also measure the kinetic properties of the single action potential. And it's fairly interesting because these properties depend upon the biophysical properties of single channels, which then can be treated as targets. For example, voltage-gated sodium and or potassium channels, different subtypes. Again, instead of injecting oscillating currents, sorry if I spent some time on the methods, but I think it's important, right? We can inject oscillating currents and see what happens of the plasma membrane. And see, by doing the Fourier transform of the voltage and of the current and dividing them one for another, getting this impedance profile across different frequencies. I said Fourier transform. This is as far as my maths literacy goes. And it's pretty cool, right? You see that at lower frequencies, you've got bigger impedance, yeah? And actually, you do have this sort of envelope that starts at a certain level, then it peaks at about, I don't know, 1-2 hertz, and then it goes down, making the neuron work a little bit like a bandwidth filter of input information. Or, if you move towards the left, the peak impedance, making it work as a low-pass filter type of neuron.
Sorry, it's
Yeah.
a millisecond?
What is milliseconds?
Yeah, it's just scale here.
Yeah, that's from. I blame my PhD student. That is 5 seconds.
Okay.
Sorry about that. So we increase the frequency between 0.5 to 20 hertz, linearly increasing within a time window of 30 seconds. So well spotted. This is a mistake. This is type. We can also measure post-firing events, which are very important, again, for the coding of spatial information in the hippocampus. This is known as the after hyperpolarization, and it has a key role in the accommodation of firing for long periods of time. And so we can measure the properties of the fast, medium, and the long after hyperpolarization. So between 0 and 500 milliseconds following the burst, and up to 10 seconds following the burst. So along the years, I worked with a number of models, which I'm not going to bore you with. I'm just going to, also because it turns out that many of these models are out of date. So I'm going to talk to you about the last work I've carried out on APP and LGF mice. These mice are characterized by the knock-in of a mutation associated with familial forms of Alzheimer's disease. So specifically 3 mutations, the Indiana mutation, the Sweden mutation, and another mutation that I don't remember. And they're interesting because they overexpress human amyloid beta, but they do not overexpress the amyloid precursor protein, which was one of the shortcomings of the first generation transgenic mice. So on these animals at 17 months of age, we have measured passive properties, so the access resistance, the time constant of the plasma membrane, and the SAG. Watch out for the SAG. The SAG is this, negative peak versus steady state type of ratio, which is associated to the activation of certain channels known as HCN channels, hyperpolarization cyclic nucleotide activated channels, which are very important for the driving of resonance properties and theta rhythm generation. But no changes in these. There is a trend to an increased SAG, but it's not significant. We also measured the average and instantaneous firing. We also measured accommodation at 5 to 10 interspike intervals, and we did not observe any change between these two. And then we also focused on the action potential waveform properties. This is the first action potential evoked, by the 300 picoamp current injection. And what we have observed is a change in the threshold, which is shy of significance. But what to me was particularly interesting here, other than the depolarization of the threshold, which may indicate an overall decrease in probability of firing, is this different shape in the phase plot of the action potential, where we plot the first derivative in time versus voltage. And you know, while for the wild types you see this nice kind of dome shape, right? For the APP you see this first acceleration, then a bump, and then a second peak, right? So this is not unknown in the field. And if you actually plot the second derivative in time of the action potential for those that show the bump versus the ones that don't show the bump, you do see that certain neurons show 2 peaks of acceleration, and certain neurons only show 1 peak of acceleration. And these peaks of acceleration are thought to be related to the difference in the point of initiation of the action potential, the axonal initial segment. So this is the action potential initiated in the axonal initial segment, while the second peak is associated to the backfiring, yeah? When the soma charges up and then it backfires, yeah?
So one thing I didn't pick up is whether the red and blue neurons are from the same population, or there is something different that it's made to them.
So this is very interesting, right? Because in the next slide, what we did basically try to quantify is the fraction of neurons within each population that had a double peak, okay?
So there is no a priori difference between these neuron types. They are neurons from
No.
the same...
Yes. Yeah, sorry, I should have probably mentioned that these are CA1 pyramidal neurons in the hippocampus, okay? So all the recordings have been carried out in the CA1 subfield of the hippocampus.
But maybe there are two types of neurons. See?
Yeah, but there could be 2 types of neurons that we don't know about, but we do know that neurons, physiologically speaking, have got different lengths of the axonal initial segment. And all sort of events can change that, not necessarily pathological, sometimes it's memory-related events, right? It's the way we respond to the environment, right? And we do know that the axonal initial segment grows centripetally. And the longer is the axonal initial segment, the closer the 2 acceleration peaks are until they fuse into one. And what we have observed overall is that in the APP and LGFs, we seem to have a higher rate of double-peaked neurons in comparison to the wild types. What does that mean for learning and coding of spatial information? We don't know. We do have network data looking at theta and gamma generation and theta pattern bursting from our collaborators at UCL. And, you know, they correlate well together, right? But it just doesn't convince me to infer causality just because it makes sense, right? And that's why I think we're missing a model in here. So before I get into that, just few results concerning the how do you call it, the resonance. We do see a left shift into our resonance, into our peak frequency, showing that, you know, the neurons tend to go from working as bandwidth filter to low pass filtering types of properties, with a decrease in the quality factor of the, or a trend in the decrease in the quality factor of the resonator. And also, you know, fairly strong impairment in the slow after hyperpolarization. So no changes in the in the medium after hyperpolarization, but this decrease in the slow one. So this is important because we do expect to affect accommodation within 10 to 30 seconds type of interval, which is consistent with what we observe in terms of phase precession of theta bursting in mice exploring a novel environment. So what? So I've got these three main phenotypes, right? And I can surely find a way for them to fit with the oscillation alterations that we observe in these animals from our collaborators at UCL, right? But is it true? Because right now, as a biologist, I don't really have a mean to do that, right? So, the hypothesis is that somehow I've got a decrease in the expression of NaV1.6 channels, an increase in expression in HCN 1 to 4 channels, and a decrease in expression in BK or IK channel, which may be causative of the three phenotypes that I observe, right? And these in turn may be causative of the disruption of network-based coding of spatial informations, specifically a theta pattern bursting and precession of the theta pattern bursting, right? I don't think I've got time to talk to you about tau pathology, but it doesn't change much. So I'm going to skip this. To get to my point, maybe this is the picture you want to take, right? This is the way a biologist think about these mechanisms, right? There is a target, and then there is these downstream effects, right? So I don't know if you've all read it, but there was a paper back in 2002, which is kind of a humorous paper, right? How would a biologist fix a broken radio, right? And then, if you haven't read it, I recommend it to you, right? So a biologist would take a radio, a perfectly working radio, right? And would probably cut a wire, yeah, somewhere here, and say, okay, so this is now the MIC, the most important component. Because if I cut that transistor or whatever that is, the radio stops working. Then another group does another experiment, and instead they remove these, right? And they call it the undoubtedly most important component, or UMIC. And after many studies, they identify this part as a target because, if they develop a transgenic radio where that component is not expressed, guess what? The radio doesn't make any sound, right? And then they come up with this model, which looks very much like mine, right? Where you've got this linear series of causalities, not just hierarchical causalities, but even temporal causalities, which evidently need to lead to the sound expression. But this is just not how organisms work, right? Because actually this is how a radio works. And so the way I'm writing grants is completely wrong, I think. Because every time I remove a gene or I knock in a gene, I'm not just removing a component, I'm also because a radio doesn't adapt, organisms do, right? So every time I put in something into it, you will have all sorts of compensatory mechanisms coming in, right? So let's talk about this, right? Because I can trust some data where I show that, I don't know, giving that drug or giving that a certain genotype can lead to, I don't know, a certain change into channel expression, right? But then how can I go from a single Hodgkin-Huxley neuron to a whole complex network and infer causality between the alteration in the single channel and the alterations that I observe in the whole network? So let's talk about this. You've got the models, I've got the data. Maybe we can make this work. So, do I have some time to talk about the Alzheimer? Yeah?
Yeah, please. 5 minutes.
Okay, so how do you run a clinical trial involving 4 universities and 1 hospital? It's like herding cats, right? So you know, one thing I wanted to try and do. No, actually, I'm joking. It was really good. Everyone worked greatly together. The mathematicians, the clinicians, myself. It was really good, right? So at some point, what I wanted to say, what am I doing with my life? I don't want to cure Alzheimer's disease in mice after I spend time inducing it, right? So. You know, I've had a chat with different stakeholders, and we came up with this idea, right, which became particularly relevant in the last year or so, because the first disease course modifying drugs have been recently approved, lecanemab and donanemab, which are relatively effective, but they're very, very expensive. And right now, the NHS has not approved their use for dementia here in the UK. They don't cover it. Okay, they're approved, but they're not covered. Also because their efficacy depends very much on the state of advancement of the disorder. And right now, a diagnosis of dementia just comes too late. You know, and if you want to diagnose it early, there are the means to do that. You've got PET amyloid to do that, but there's no way you can screen population by doing PET amyloid in people that don't show any symptom, any cognitive symptom. So there is scope to develop non-invasive and low-cost early diagnostic methods with a certain predictive capacity for Alzheimer's dementia. So EEG is not new to this. There are countless studies trying to use EEG as an early predictive tool for dementia. There are fewer studies looking at the prediction of MCI converting into dementia, and even lower looking at the prediction of, you know, cognitively healthy people converting at 2 years, 5 years, 10 years into dementia. And the problem is, you know, the overall low sensitivity, and so far, the truth is that there's been lower demand. Because every time you were applying for a grant, you say, okay, fine, you want to diagnose early Alzheimer's disease, but then what are you going to do with that information? You can't cure it anyway, right? There's not a cure. But now there's not a cure, but at least there are disease course modifying interventions. Anyways, we came up with a study in collaboration with the University of San Marino and the Department of Neurology in San Marino, and we recruited 41 people, 10 with Alzheimer's dementia, 10 with single domain amnestic MCI, and 11 without cognitive deficit. And then the people with MCI has been followed up for 2 years until 10 converted into full-blown dementia and 10 stayed at an MCI level. And in each one of them, we've carried out a battery of cognitive tests and open and closed eyes resting state EEG. And starting from the battery of tests in terms of scoring different neuropsychological functions, we have observed that the stronger effect in predicting whether or not one would develop dementia, would convert into a dementia state, was the Stroop color word test, and especially the number of errors. We've seen a significant difference across the years between those who would convert versus those who would not convert. Then we would take the whole power spectrum of these patients and build a model of this whole power spectrum. Don't ask me how, they know. Dominic and Mark know how they did that. And the interesting part, I think, emerged when we were comparing the alpha peak between converters and non-converters, and then seeing that actually the single spectra of each one of these patients was very, very different from one another. So rather than grouping these into populations and splitting them into bands, what we've decided to do is to use a different approach, an unsupervised k-means clustering type of analysis, where the whole spectrum has been compared to a centroid and identifying, putting as a restraint number of 3 groups and identifying therefore 3 types of clusters, and then counting who was included in each one of these clusters. And interestingly enough, what we have observed is that cluster 1 contained all the healthy people and the vast majority of the non-converters.
How big is the sample? It was like around 20 people?
20 people, yes.
But isn't that a small sample?
Very small sample. That's why we can't publish it. Such a great study, but we keep being kicked back for the thing. So it is small, but we've got a very clear hypothesis and a fairly biased population. So yeah, is it generalizable? Absolutely not. Does it grant further, well, not necessarily, we don't know yet, but does it grant further investigation? Absolutely yes, I'd say. Also because we have observed, just by using, the clustering and the neuropsychological scores, we would get a certain level of accuracy, but actually combining them together, we would get a nice level of area under curve. Of course, with the risk of overfitting, right? Because we don't have too much numbers. But I think that for the type of measurements that we've carried out, it was a reasonable number, and all the power calculations we've done to test the hypothesis that we've tested showed that our sample, based on the effect size and the variance, was big enough. So for the limited hypothesis that we've tested, the sample size was big enough. But we need to be careful in generalizing it, yes, for sure. So the conclusions are that, I didn't show you the data because I don't have time, right? But we also shown that these observations correlate. I know I'm late, but they asked some questions, so.
No, you're fine.
I'm fine. Some of these phenotypes actually correlate fairly well with an impairment in attentional, in an impairment in connectivity in the attentional executive network in the brain, and also with decreased excitatory and increased inhibitory drive as modeled using these data. So yeah, thank you very much for listening. Thanks to my collaborators, especially Sofianna, my PhD student, for carrying out all the measurements, Daniel, postdoc I share with Wendy, and of course Reading and San Marino and IR UK for funding the research. Thank you very much.
Go on then.
I have a question and a comment. Can I start with the comment first?
Of course.
Now your idea of the actual initial segment, the IS, and coupling with the instrument, that dictates that change in the AP cycle when the DVDT versus replay.
Yeah.
It's been actually studied. There's a paper using Hodgkin-Huxley models that couple these two compartments together to show that phenotype in defining shape. So you're in the right track. That's what I'm trying to say, is that there are studies that have been done that's done this thing. Now my question is...
nice.
Nice. God, I forgot my question now. Oh yeah, when you showed the chirp.
Sorry, the what?
The chirp. You know, the stimulus with the sinusoidal wave stimulus, where the frequency is increasing.
There is
yeah. So you said at one point that it's acting as a bandpass filter, but your your simulation, your data is showing that is acting as a low-pass filter rather than a.
Well,
Am I missing something?
no, you're not. Okay, so the population works as a bandpass because your peak impedance doesn't correspond to the lowest frequency that you're injecting, right? But you're right, it could be a better bandpass filter. It really depends on many factors. The truth is that I'm going to show you the example trace because it shows it a bit better, right? Here. So, yes, what you do see is that you've got peak impedance at about here, between 2 and 5 hertz, right? That's why we say it's more of a bandwidth filter. While, when you've got, but you're right, like sometimes you have it here, depending on the neuronal population, sometimes it moves left, and the more left it moves, the more it works as a low-pass filtering system. So what you care about is the difference, really, right? If you've got an event, physiological or pathological, where it moves, that impedance. And if you look at the impedance profile, the peak frequency is around here, which is not at the lowest level. But yeah, you're right, they could resonate better.
Quick one from Daniele, I think that's the last one.
So I'm contacting you with regard to the maybe I can help slide. So can you comment a little bit about the sort of network size you're thinking about? I'm asking this because I'm conscious that if the network size is large enough, there is no model in existence that can make you have conductances that you would want to describe macroscopically to appear at macroscopic level. Whereas if your network size is a modest size, then tell us what sort of effect you're expecting.
Okay, so I am a big fan of bottom-up approaches when we're looking at these type of phenomena, right? So to say, rather than going from a network alteration, for example, the network alteration that we observe in the APP and LGFs is this disappearance of the phase precession of the theta burst firing, of the theta pattern burst firing of place cells, right? And what we usually do in the field is to then use this top-down approach where we try to kind of rationalize, justify the network behavior by finding channel alterations that could explain that, right? I think we're already biasing ourselves that way. I would like an approach that works the other way around, right? Where I take conductance information from CA1 pyramidal neurons, different types of interneurons, and the strength of synaptic connection between these populations. And these are data we've got.
Can you tell us if n is the size of your population? Can you tell us if n is of the order of dozens or millions?
It's not millions, it's not dozens, it's hundreds. Yeah, so we're talking about hundreds of recordings. Across different models, thousands, but within a model, we're talking about between tens and hundreds of recordings on a good project, so to speak, right? Hundreds when we are considering the combination, not hundreds for each cell type, but the sum of excitatory pyramidal neurons, inhibitory feedback interneurons, inhibitory feed-forward interneurons, and these sort of things, right? So, for example, I've got a study on TG4510 tau mice, where I've measured the effect of tau at different time points on OLM interneurons, neurogliaform cells interneurons, and CA1 pyramidal neurons. It is a monstrous job. It's just a lot of work. And I would like to explore if I could take those data and put them into a model that connects the Hodgkin-Huxley to a network and say, if I change the conductance of HCN from such to such, what happens to the network? And then compare what I observe in real experiments versus what the model gives. Does that make sense? Does that answer your question?
Yeah.
But why would a model of interconnected neurons be sufficient? So where are the other cells? Where are the other modulators? Where are the connections from other regions? Like how does this, I guess going back to your radio analogy,
Yeah.
how does this let us understand the in vivo system?
No, you're right. You're right. The good news is that for the hippocampus, the inputs are quite well defined.
The synaptic inputs?
Yes.
What about other neuromodulatory networks?
I don't know.
Well,
I don't know.
well,
I
if
just-
you keep everything as is and you alter one parameter in a model,
But,
you could see the effect of that parameter on, like the HCN conductors, for example.
yes.
Yeah, but
Everything stays the same, and then you change one at a time. This is at the end of the day is a model. It tells you what's the impact of this on the network.
yeah, but the brain is not a network of neurons. There are other transmitter systems, there's
No, but-
neuropeptide systems, there are other cells.
But I agree with both, right? I think having a reductionist approach is correct as long as it's a cumulative reductionist approach where you keep adding ingredients to the network until, you see what I mean? And I think also culturally is an important thing, right? Because I think we are very biased in rationalizing the macroscopic observations that we have, and we never play with fundamental data and test the quality of our models to see what kind of predictions they give us. And we do have a lot of data in that sense. And then maybe that would help that question, right? Because the moment we see that there is a discrepancy between the observed output, the observed phenotype,
Yeah.
then we can just come in with other components. Right?
Discrepancy, yes, but it's where it might be misleading. So, for example, a change to a certain ion channel conductance allows you to replicate the data, but in reality, this was driven by something non-neuronal, and then you're on a path that isn't the truth.
You're right.
Yeah.
You're right. But I guess that's where the experiment comes in, right?
Yeah.
So you've got the first experiment to get the data, then you run your bottom-up model, that informs a hypothesis, and then you go and test that hypothesis experimentally. That's just something we don't do. You know what I mean? Like we come up with new hypotheses assuming this linear sequence of causes. It's even worse, it's the worst way we could do things. Right? So maybe it's not going to be perfect what we come up with together, right? But it would definitely be better than, A does B that does C that leads to the disease. So if I cancel B, I cure the disease. Like, this obviously doesn't work, especially in Alzheimer's disease.
Is it a quick one, Goran?
I hope so. Can you go back to that slide where you did the clustering of the
Sorry?
Can you go back to the slide where you did the clustering of the EEG?
Yeah.
That was predictive of the MCI.
Yeah.
I just want to make sure that I understand.
Yeah.
So you had.
It's about the similarity of the spectra.
Yeah, no, I understand that part. And then, so you were. I'm just trying to understand the subject. So you were clustering the EEG according to some the spectrum, and then the cluster number one was the patients that they were the normal people, right?
So in cluster one, there are a certain number of people, right? And so the proportion of these patients in the cluster one are the totality of the healthy ones are in cluster one. But in cluster one, there are also non-healthy people.
I understand, but these were not the same patients like sometime later.
No.
They're all at the same time, or roughly at the same time, and some of them were healthy according to what? Like some MMSE or MoCA score, they were above some
Yes.
score?
Yes. So the inclusion criteria for cognitively healthy people was people that didn't show any sign of cognitive decline at the MoCA. But for inclusion criteria for Alzheimer's dementia and MCI is that for the Alzheimer's dementia they needed to be positive to cerebrospinal fluid amyloid beta and phospho-tau levels, and they had to show severe cognitive decline, multi-domain, while for the MCI group, they needed to show single domain amnestic MCI to a battery of tests that has been carried out, testing attentive, executive functions, short-term memory, and long-term memory.
I see. So would you think, or do you have any evidence to suggest that your test, as you have it right now, and I understand you don't have a lot of patients or something, but do you have any evidence to suggest that your test is more sensitive to, in particular, early parts of dementia, like MCI, compared to the MMSE and MoCA, which have clear deficiencies in identifying MCI?
Yes, so this is what we think, right? Especially based on the fact that people that are considered MCI and not converted actually have got severe impairment in their attentional executive functions, which currently are not considered for a diagnosis of Alzheimer's disease. And the EEG trace, by using, don't remember what type of model, but I'm going to tell you, shows that also the connectivity of the prefrontal to the temporal, the areas involved in attention and executive functions is impaired in these patients. And then curiously enough, I had a chat with a clinician, and they said, but everyone knows in the hospitals that people with mild cognitive impairment that have got poor executive functions will convert into dementia. I was like, who's everyone? Because in literature there's zero. It's just a thing. We know.
Brilliant, thanks Francesco.
Thank you.